Hello, my name is Justin Filardo, and my talk today is titled No Silver Bullet: Problems with the Use of qPCR in Cannabis Safety. So, as I said, my name is Justin, and I'm a PhD candidate in the Wong Lab of Molecular Food Safety at the University of British Columbia. And my specialty is in the food safety of soft ripened cheeses like Brie and Camembert. You can say I lead a very delicious life. But you may be wondering why I, a food scientist, am here today to discuss microbial analysis in cannabis. And it's not just because cheese is a delicious snack when you're high. It's because cannabis safety is food safety, particularly from a microbial perspective. Put simply, we are both interested in isolating and/or enumerating key microorganisms from complex matrices. So it wasn't a stretch when Willow Analytics hired me to investigate the use of qPCR for microbial enumeration in cannabis. As a quick refresher, microbial enumeration has been traditionally done through culture-based plate counts, shown at the top. The cannabis sample is first homogenized to release the microorganisms into suspension. Then this microbial suspension is serially diluted and plated. After a suitable period of incubation, the number of resulting colonies, or colony-forming units, or CFUs, gives us a reasonable approximation of the number of target microbes in the original sample. The drawback is that this method can take up to 5 days depending on the microbes you are enumerating. The qPCR method, on the other hand, shown at the bottom, provides an attractive shortcut to this lag time, with results generated in as little as 1 day. First, the total DNA is extracted from the microbial suspension. Then the DNA is analyzed using a suitable qPCR assay, generating an amplification curve. The amplification curve is generated through the amplification of a DNA target, as shown on the left. qPCR uses a pair of primers targeting a particular gene or DNA region, causing it to duplicate itself. If that particular DNA sequence is present in the sample, then at each qPCR cycle, the number of amplicons, as they are referred to, doubles, causing an exponential increase over the course of the qPCR reaction. And using a fluorescent dye, we can monitor the increase over time of these amplicons, leading to the resulting amplification curve shown on the right. The best thing about this is that the cycle where the fluorescence passes a specified threshold called the CQ value is proportional to the starting concentration of the DNA target, putting the quantitative, or Q, in qPCR. As an example, here on the left is a qPCR reaction of decimal dilutions of two different bacteria, one in red, one in blue. As you can see on the right, in both cases we get strong correlation between the CQ value on the y-axis and the log concentration of microbial cells determined by plate count on the x-axis. So it's no surprise that there has been a lot of excitement about using qPCR to rapidly quantify total bacteria and total yeast and mold in cannabis. At least until this happened. Earlier this year, Michigan regulators, citing an unacceptable level of false negatives, put a temporary ban on the use of qPCR for total yeast and mold, even warning that moldy cannabis products could have made their way into the market. So here at Willow, we wanted to do our own full laboratory investigation of multi-species enumeration using qPCR and have prepared a white paper describing our results. Spoiler alert, this technology is not the silver bullet it appears to be at first glance. I don't have time to present all our data here, but I would like to highlight some of our key findings with respect to variation between microbial species and correlation to plate counts. I encourage you all to check out our white paper for a more detailed analysis of findings. For a broad species qPCR assay to be suitable, it must meet these three criteria. First, it must target all members of the group being quantified. Second, it must show consistent amplification between all those members of the group. And finally, it needs to correlate with standard methods, in this case, culture-based plating. To test the first two of these suitability criteria, we compared a variety of universal primer sets for bacteria or fungi against a variety of microorganisms in pure culture. We also included a commercially available qPCR assay for both total aerobic count and total yeast and mold. The cultures were serially diluted, and each dilution was enumerated in parallel by both plate count and by qPCR. Of the universal primer sets we tested, only the 16S ribosomal RNA and ITS primer sets showed suitable results across all bacteria and fungi, respectively, so this is the data I will be presenting. For clarity, I will refer to them from here on simply as the TAC assay and the TYM assay. For comparison, I will also share the data from the two commercial kits. Looking at our results across our 4 bacteria using the TAC assay, we can see that there's a strong correlation between qPCR results on the y-axis and plate counts on the x-axis. This was expected. It shows that 1, the assay is functioning properly, and 2, I didn't screw up the experiment, which is good news for me. The issues arise, however, when we look at all 4 bacteria together. On this graph, you can see that there can be a greater than a half log difference between the different species at any particular CQ value. To put this in perspective, the same result by qPCR could mean 50,000 CFU per gram or 160,000 CFU per gram, depending on which species are present. Put simply, based on compendial guidance of 100,000 CFU per gram, this means that the same qPCR result could mean both a pass and a fail, depending on what organism you're using for reference. Similar results were observed for the commercial kit, suggesting they are also targeting the 16S ribosomal RNA gene just like us. This discrepancy between species was even greater when enumerating total yeast and mold, with more than 100-fold difference between yeast, shown in blue, and mold, shown in red. That's the difference between 10 and 1,000 CFU, both with the same result by qPCR. And an even greater difference was observed with the commercial assay shown on the right. There's no way that meaningful results can be generated with this sort of interspecies variation. Now, there are a couple explanations for these discrepancies, but the most important is that cells does not necessarily equate to DNA. The ribosomal DNA, which is the most common target of universal primers, has varying numbers of copies across species. In bacteria, on the left, we can see a range of 1 to 13 copies, while in fungi, on the right, it appears to range from 25 to 250 copies. This is a 10-fold difference across the groups we're trying to accurately enumerate. Put simply, this means that consistent conversion of CQ to CFU across a multi-species community is highly unlikely. And even if we could find a single copy universal target, we need to further acknowledge that CFU does not equal cells. Cells exist in clusters and chains, meaning a CFU rarely results from a single cell. This is especially true for molds, which are themselves multicellular organisms. But we weren't going to let that stop us from seeing how these assays functioned in a real-world situation. To do this, we compared our 4 assays against traditional plate counts in 6 plant samples: 2 hemp, 2 irradiated cannabis, and 2 non-irradiated cannabis. As you can see, we found very little correlation between qPCR results and plate count results, with similar CQ values for samples showing both high and low concentrations by culture-based methods. And the commercial kits, shown at the bottom, even produce contradictory results with an upward sloping trend line as opposed to the downward trend expected from qPCR. These results highlight a third issue with using qPCR for microbial enumeration, that of dead DNA. Throughout the drying and/or irradiation of cannabis, much of the viable bacteria is expected to die off. Unfortunately, those dead cells still leave behind their DNA, meaning that qPCR will enumerate these dead bacteria as if they were still present and viable. This means that even irradiated cannabis with no viable microorganisms fail a qPCR test. So in closing, qPCR sounds exciting and is a great tool for the detection of specific microbial pathogens, but is not going to work for broad species enumeration in cannabis. Variation in the results across different microorganisms makes reliable enumeration impossible, and a lack of correlation exists between plate counts and qPCR in the cannabis matrix. Finally, I want to point out that no qPCR assay for broad species microbial enumeration has yet been third-party validated by an accredited organization such as AOAC, not just in cannabis, but even in the food safety arena. So I think it's safe to say that qPCR is no silver bullet when it comes to cannabis safety, and producers and labs should think twice before relying on this technology for microbial enumeration. With that, I would like to thank all my colleagues at Willow Analytics, especially Emily and Jay, who helped with the technical work, and Jerry for his support on the project. I would also like to thank you all for your attention today and remind you to go check out our white paper for a more detailed discussion of the results presented here. For a copy, you can contact us directly at willowanalytics.com. And now I would be happy to answer any and all questions you may have.